So without much ado, I will move on to the first speaker. I'm very pleased to be able to introduce my colleague, Professor Ultan Power from Queen's University, Belfast. Ultan is Professor of Molecular Virology in Queen's in the Wellcome Wilson Institute of Experimental Medicine. He graduated from University College Cork with his PhD in microbiology, and then he went and did his postdoctoral training in the molecular virology of respiratory viruses in St. Jude's Children's Hospital in Memphis, Tennessee. Ultan then moved into the pharma sector in France, where he was appointed head of virology in the Center for Immunology in Pierre Fabre, which is the biotech research arm of the Institute to Research Pierre Fabre. And there, Ultan played a central role in conception of research and development of the first subunit vaccine against respiratory syncytial virus, which entered into phase 2/3 trials. So Ultan is what he's going to talk to you about today. In fact, both speakers, this is talks are around SARS-CoV-2. So this is kind of a new direction, which a lot of people have taken during the actual outbreak. So Ultan is actually going to talk today about their work on drug repurposing, so SARS-CoV-2 and drug repurposing. Over to you, Ultan. Thank you.
Thanks a million, Louise. And
sorry, go ahead. Can you share your screen, please?
I can indeed, yeah. Okay, so that working okay?
Yeah, just put it on full screen, yeah.
There you go.
Thank you, perfect, thank you.
Okay, well, listen, thanks again, Louise, for the invitation, and to the UK-ICN for the invitation to be able to talk to you a bit about the work that we are doing. Hopefully I can get this working all right. Bear with me. There you go.
Okay.
So yeah, as Louise mentioned, I'm going to be talking about some of the work that we've done over the last two and a half years or so on the drug repurposing and the rationale for it, why we're doing it, and where we're at with it at the moment. Just give you an update with this particular project. So this is one of the first projects that was funded through the NIHR UK-RI rapid response call for SARS-CoV-2 or COVID-19 pandemic, as it were, at the time. And really the goal and the rationale behind trying to use drug repurposing is just based on the possibility of being able to be much faster in terms of the speed with which we can identify drugs and get them into clinical trials as quickly as possible. If you just look here, with the normal from discovery to actual marketing of drugs, it often can take up 10 to 15 years to be able to get a brand new drug through the whole process of research, development, and then subsequently clinical development and marketing. However, with repurposed drugs, the idea is we use drugs that are already either on the market or have been through clinical trials already. So their toxicity profiles and their profiles in humans are already well known. And basically what we're doing is taking those drugs and asking the question, have they any capacity at all to be able to block viral replication or infection, as the case may be, and therefore because their safety profiles are well known, the speed with which we can get them into clinical trials is accelerated substantially. So now we're going to, within 2 years, we hopefully will be able to get products that through the market. And that's proved to be the case over the last couple of years in terms of some of the drugs that have been tested to treat COVID-19 patients. Notable example would be dexamethasone, which is one of the very early drugs that was proven to be successful in treating the actual disease itself. So this is the rationale for the drug repurposing as opposed to de novo drug discovery. Now, the rationale for the actual project itself, our thinking was that SARS-CoV-2 primarily targets the airway epithelium, but we also know that this viral RNA in many tissues detected, and in fact, viral proteins detected in many tissues throughout the body. The inflammatory process is probably the main process associated with the pathology associated with COVID-19. So therefore, we anticipated that if we could find drugs that targeted the airway epithelium, we would have therapeutic benefit, either from an anti-inflammatory point of view, but what I'm going to concentrate here are the discovery that we did, or the screen that we did for antiviral activity. And our goal, as I say, was to screen bespoke, FDA-approved or other approved drugs that were safe in human for antiviral activity against RSV in our in vitro systems, and I'll explain those to you as we go along. So before we started, obviously, this is what most people did at the very start of the SARS-CoV-2 epidemic. It asked the questions, which cells were the most susceptible to infection and therefore the most appropriate for looking for antiviral activity. And this is what we did as well here, showing a range of different cell lines that we tested for SARS-CoV-2. And what we found was that Vero cells and Calu-3 cells were probably the best in terms of the amount of virus that was capable of replicating from these viruses and the infectivity of these particular cell lines. And these are the ones that we chose to continue on to work with in our screening profiles for these viruses. Now, the actual drug library itself contained over 2,500 drugs. It was made up of an FDA-approved drug list that we had, an antiviral drug list, which was about 50%, and then a drug list from virtual screening that was done just around the time at the start of the pandemic, which we included in our overall drug screen. And how we actually did that, the initial high throughput screening was by drug spotting on 384 well plates, 24 hours before seeding the cells with, seeding the plates with the cells, either the Veros or the Calu-3s. The day after, we infected those cells with SARS-CoV-2. Again, we did quite a bit of work to optimize this protocol to make sure that we had sufficient readout at the end of the experiment. And then 3 days later, we're looking for cytotoxicity, cytopathogenicity of the cells following infection with and without the absence of the drugs. Okay, so this is what we're able to do, and then screen those cells cytotox screen assay to allow us to determine whether or not there was any evidence of cytotoxicity, which we would expect to see in untreated cells, and then how much was that reduced following treatment with the various drugs that we looked at. In the initial screen, we were able to look at 10 drugs per well, and then subsequently we deconvoluted those to either 2 drug combinations or single drugs, as the case may be. And all I'm going to talk to you today is about the single drug screening that we've actually ended up doing. Now, this just gives you an overview of the drugs. If we have an 80% cutoff in terms of a reduction in cytopathogenicity here, we found that in the Calu-3 cells, we identified 22 drugs that were positive, had antiviral activity, and that is reducing CPE by 80% or more. And we had 33 drugs that were positive or had a positive outcome in the Vero cells, okay? And this is to say we're screening 5 micromolar of the drugs, SARS-CoV-2 with a multiplicity of infection of 0.1 and looking at for CPE at 72 hours post-infection. So this is again showed that, and this is consistent with a lot of the repurposed drug screening that has been published since then, showing that a tremendous amount of the drugs fail. It's very few of them have actually shown any significant antiviral activity against SARS-CoV-2. So of the drugs that were there, we came across most of the drugs that were screened. We took 40 drugs on for further screening, and we found that 28 of them could be eliminated because they had insufficient activity when we did dose responses. They had insufficient antiviral activity as we lowered the doses from 5 micromolar. And at the highest doses, some of the drugs were actually cytotoxic. So we were able to eliminate quite a few of those quite well. 3 drugs we found were highly positive. They're 100% effective in both cell lines in a reasonable dose range. This would be 0.67 to 4 micromolar, okay? And 2 of those have already been described. These are licorine hydrochloride, which is a plant extract, which is an RNA-dependent RNA polymerase inhibitor, and another molecule called GS441524, which is a nucleoside analog, not unlike remdesivir, and developed by Gilead. Okay, so both of those drugs have proceeded to clinical trials. The third one from this group that we found was interesting, and it's the one I'm going to talk about for the rest of my talk, is azatidine. And when we looked at azatidine in terms of cytotoxicity, again, even at the highest concentrations, there was no evidence of cytotoxicity associated with this drug. And when we looked at the antiviral activity, we were determining an EC50 of 4 micromolar, which was, as you can see, is higher than the EC50 from remdesivir. So in many cases, we use remdesivir as the positive control because this is the antiviral that has been used in clinical therapies against SARS-CoV-2 or COVID-19 to treat high risk or highly vulnerable patients in the clinic. So the safety profile of azatidine looked very good, and the antiviral activity, while it wasn't as effective as remdesivir, was nonetheless quite encouraging for our initial studies. Now, in order to take this further, obviously, we wanted to look at SARS-CoV-2 growth curves and the impact of the virus, the antiviral activity of azatidine on those growth curves. Now, this is just showing you typical plaques that we have identified with SARS-CoV-2, and this is looking at a growth curve in viral cells starting with an MOI of 0.01. So this is a typical growth curve that we would see. Now, when we looked at the ability of azatidine to block virus infection, as in productive virus infection, again, the cells were treated with either 5 micromolar or 25 micromolar of azatidine, and in all cases, we see a massive reduction in the amount of virus present at 72 hours post-infection. And similarly, if we looked at qPCR for NSP12, again, we found at 25 micromolar virtual elimination or undetectable levels of genome copy numbers by 72 hours post-infection. Again, similar to what we're seeing with 1 micromolar remdesivir. So it looks very, very encouraging indeed. Importantly, when we combined azatidine and remdesivir, we found that we were getting a shift to the left in terms of the EC50 concentrations. Here you have in blue looking at azatidine alone. We find, as we found before, an EC50 of approximately 4 micromolar. But when we combined remdesivir and azatidine, now we find this shift to the left and a reduced EC50 to 2 micromolar, indicating that these drugs were complementary, and in fact, our evidence suggests that they are synergistic and work synergistically together. And similar, if you look at remdesivir, this is blue, you have remdesivir alone, an EC50 of 0.6 micromolar, but when we combine both, we find we're getting a shift to the left, where now the EC50 of remdesivir is reduced to 0.25 micromolar. Again, evidence of synergistic interaction between these 2 drugs. Now, one of the significant ways then to look at the antiviral activity, all of the drug screening we've done so far was done where the drugs were treated prophylactically onto the cells before viral infection. So that obviously doesn't reflect what's happening in the clinic where people come in and they're already sick with the virus. So in order to replicate the virus, the clinical situation, we've also set up an infection model based on well-differentiated primary airway epithelial cells. Okay, this is a model that we work a lot with in my lab, working primarily as I do with respiratory syncytial virus. But what we found is we did quite a lot of work in setting up this model where we infected the cells with SARS-CoV-2, looked at the various growth kinetics, inflammatory responses like cytokines and chemokines, etc., just to set the baseline levels of how SARS-CoV-2 interacts with airway epithelium. We also determined that when you infect the cells with SARS-CoV-2, they don't create a huge amount of damage, unlike, say, in monolayer cells, where the cells are wiped away within 72 hours, depending on the multiplicity of infection that's used. But in primary airway epithelial cells, they're actually fairly resistant to the virus. So it meant that we could set up models whereby we could infect with SARS-CoV-2 and then come back in 24 hours later with the drug to determine whether or not the addition of the drug in a therapeutic model was capable of influencing the outcome, subsequent outcome of the virus growth kinetics and inflammatory responses to infection. And so this is what we've done here. Infected with SARS-CoV-2, treated with the drug, either remdesivir or azatidine, and both in combination as well. And then at 72 hours post-infection, we did, we actually all the way through every 24 hours from the 24-hour time point right to 72 hours, we did an apical wash to determine the amount of virus that's been released from the cultures. And at 72 hours, we did an RNA extraction to look at the amount of virus that's resident genome copy numbers that are present in the culture following treatment with the drugs. And these are the data we came up with. In blue here, you have the untreated or DMSO controls showing the normal growth kinetics that we would expect for SARS-CoV-2 in these cultures. Now, this is the N = 5 donors, so it's a pretty comprehensive data set here. In red, we have azatidine treated, so infected and then treated with azatidine. And what you can see here is that azatidine is having a dramatic impact on the growth curve of SARS-CoV-2 in these cultures. Okay, so dramatically reduced compared to the untreated controls. As expected, remdesivir was more efficient than azatidine, but when we combine both remdesivir and azatidine, bearing in mind that we're now dealing with 2.5 times less drug for both azatidine and for remdesivir, we get at least equal, if not superior reduction in virus growth kinetics when following treatment with both drugs compared with either drug alone. Again, confirming in a therapeutic model that azatidine, either alone or in the presence of remdesivir, has a very significant impact on SARS-CoV-2 replication. And similarly, when we look at 72 hours for NSP12, we find there's a virtual elimination of the virus from the cultures by that time period. Okay, again, very, very encouraging results, to say the least, in relation to the possibility of using azatidine as an antiviral against SARS-CoV-2. But similarly, when we look at the inflammatory response, again, this is looking at the RNA level, and we've looked at interferon lambda-1, interferon beta-1, IFIT-1, ISG15 or IL-6, also IL-8, CXCL8. We find that there's a huge reduction in the inflammatory response, at least for 5 of these genes. Okay, I'm not overly surprised with IL-8 because we know that in these cultures, the endogenous level of IL-8 expression is very high, even without any stimulation. So it seems to be an endogenous level of expression of CXCL8, IL-8. But all of the rest of them are showing a very, very significant reduction in the inflammatory response. And bear in mind, this is in a therapeutic model. So the virus has already had the opportunity to replicate for 24 hours before treatment. So again, very encouraging from potentially from a therapeutic point of view. Now, what is azatidine? Azatidine is a first-generation antihistamine. This was developed by Shiri Plow and put on the market in 1973. It's a typical antihistamine, as I say, first-generation antihistamine, used to treat sneezing, runny nose, etc. So I'm sure anybody that suffers from allergies will be well aware of the signs and symptoms associated with that. And these are the doses that are normally being given to people to treat allergic type of reactions. However, it's been heavily associated with drowsiness and therefore was discontinued and superseded by loratadine and desloratadine. Okay, so these are newer drugs that have been designed to eliminate or diminish the drowsiness associated with this first-in-class drug. Now, is there any evidence at all of any other antihistamines? I should point out that we also screened loratadine and 28 other antihistamines, and azatidine was the only one in our hands that showed antiviral activity against SARS-CoV-2. But is there any evidence anywhere at all to back up the idea that azatidine might be useful? And this is some evidence from in silico screening suggesting that azatidine has the capacity to interact with the 3C Pro, and so therefore it possibly is working as a protease inhibitor. There is some evidence from other screens as well, masitinib, for example, showing antiviral activity against SARS-CoV-2. So this is another antihistamine that has suggested or shown to have some antiviral activity. And then in the clinic, there is a famotidine, which again, this was looking at a clinical study of people who were on antihistamines, this particular antihistamine versus those that weren't. And there was a suggestion of reduced fatality associated, or hospitalization, associated with our patients that were receiving famotidine compared to untreated controls. Again, suggesting some indirect evidence that maybe antihistamines might have a role to play in the war against SARS-CoV-2. So just to conclude then on this, azatidine has therapeutic potential against SARS-CoV-2. It acts synergistically with remdesivir, and it may be suitable for early drug use in view of the fact that it was used as an antihistamine over-the-counter drug. It may actually have use early on in the antiviral process. So basically, when somebody becomes confirmed positive or test positive for SARS-CoV-2, this could be a drug that could be used early on in the process to diminish or help prevent individuals, particularly highly vulnerable individuals, proceeding to hospitalization. And this fits then with the UK antiviral task force priorities for antiviral drugs against SARS-CoV-2. So this then for us also provides a strong rationale for further development of azatidine. Now, I just want to finish up very briefly then with a little bit of work that we've done with monoclonal antibodies. So as you know, monoclonal antibodies were another area that have been developed rapidly for SARS-CoV-2 therapeutics. And the one that probably led the race for the longest time was Ronapreve, which is a combination of two antibodies in imab and casirivimab. Okay, so we then wanted to look and determine, with the advent of the new variants of concern, what role are these playing, or do they have any potential role in antiviral activity or therapeutic possibilities for individuals who are hospitalized with SARS-CoV-2. So we looked at antiviral activity in a virus neutralization assay for Ronapreve, imdevimab, casirivimab, and sotrovimab, which is the other one we managed to get hold of. There are two other monoclonal, either combination or single antibodies that are currently being used, including Evishield, which has an altered Fc domain, which gives it a much longer half-life than normal antibodies. And this particular drug combination is suggested to be used for individuals who are at high risk of severe disease, but for whom their immune system is not working very well, and therefore they might be able to provide some protection against SARS-CoV-2 infection. So in our hands, this is just some data looking at the viral neutralizing activity. The lower the number here, the stronger the antiviral activity. And when we looked at alpha, we found that the Ronapreve and the individual components of it were quite efficient at blocking alpha and delta. Sotrovimab was not any good at blocking alpha, but very good against delta. It was thought to be the savior in the advent of the Omicron era, but in our hands, it worked quite well for Omicron BA1, but lost activity against BA2. Again, leaving us in a situation with the Omicrons that there are very few options available for that. There is some evidence to suggest that Evishield still maintains antiviral activity against the Omicron variants, but we haven't tested that specifically ourselves. So the outcome of this, from my perspective, is that we need to pay a particular attention when we want to use and prescribe monoclonal antibodies. My suggestion is that individuals, the virus that is affecting a given individual needs to be sequenced before using particular monoclonals, because, for example, giving Ronapreve or sotrovimab to BA2 would be like absolute waste of time from a clinical point of view. So I think there's some coordination that's going to be needed to sequencing viruses in real time in hospitals to be able to facilitate the prescription of these monoclonals. Now, the last thing I just want to finish up here is just we were successful recently in obtaining a DPFS funding to continue on this work to repurpose approved drugs as potent antivirals. And this is going to continue on the work from a combination point of view. This is work that a project that we're leading from Belfast, but it's in collaboration with the University of Liverpool, James Stewart and Andrew Owen, and also the University of Oxford with Miles Carroll. Basically, what we're going to be doing here is we will continue on the high throughput screening and the in vitro models for the antiviral activity combinations. Liverpool will have responsibility for in vivo models and pharmacometrics. And Miles Carroll in Oxford will be looking at drug resistance and escape mutants to determine for any of the drugs and drug combinations we identify, what is the propensity for those to develop antiviral resistance, okay? So that's an ongoing project that just started in April 1st this year, and hopefully we'll be able to tell you more about that as the project progresses. Just like to finish up then very briefly with thanking the individuals who were responsible for this work. In particular, Alan Athley is a postdoc in the lab that came up with the original idea. Her background is in drug screening for leukemia, and she came up with the idea that maybe we combine the drug screening capacity and expertise that they have in our Cancer Research Centre with our antiviral activity in the virology department. And this really was the genesis of the project. And then the various people who helped work on this project over the last year and a half or so, 2.5 years, I should say, with David Courtney, Lindsay Broadbent, Connor Bamford, Shireen Mansour, Olivia Toussaint, and obviously this was done in collaboration with Ken Mills in our Cancer Research Centre. And in the regional virology lab in Belfast, Conal McCaughey as well has been absolutely fantastic. One of the major things as well that happened at the start of the pandemic was the level of collaboration, the level of data sharing, and the level of interaction there has been across the UK in particular, but across the world actually. And this has been absolutely phenomenal. And I certainly would appeal to everybody to maintain this level of collaboration and interaction because we've shown, I think, collectively, we've shown that we can do an awful lot of really good work in very, very short period of time if we have the right investment and the right collaborations. And again, I'd just like to thank the folks who loaned us some equipment to help us progress the projects, and then the individuals who provided significant advice, protocols, and reagents at the start and continue to do so throughout the project as well. So again, for the individuals that are named here, thanks a million for all of the help. And obviously, none of this work would be possible without the funding from the UKRI in our case, but also the HSC Public Health Agency in Northern Ireland as well, who provided significant funding. Okay, so I'm going to leave it at that. I'll stop sharing my screen and very happy to take any comments.